Hi, everyone. Arthur and RF here with Endpoints News, and thanks for joining us today for Ensuring Sample Integrity from Collection to Analysis. We're sponsored by Agilex, and I'm excited to moderate today's expert panel. Joining us today, we have Jason Goy, the Research and Development Manager, Mass Spectrometry at Agilex Biolabs, and Jean Ray, the Senior Scientific Advisor at Agilex Biolabs. If you have any questions during today's webinar, our panelists have reserved some time for questions at the end. So just hit that Q&A button at the bottom of your screen as soon as you think of a question. This webinar will be available on demand tomorrow to rewatch or to share with your colleagues. Now I'm going to pass it off to Jason Goy to get us started. Jason.
Thanks, Arsalan, and thanks for joining. My name is Dr. Jason Goy. I've been at Agilex for 10 years. My background is in mass spectrometry. Our team develops assays for small molecules up to peptides, say 8 kilodaltons large. Agilex Biolabs headquarters is in Adelaide, South Australia, and my co-presenter is based in USA, and I'll let him introduce himself.
Thanks, Jason. My name is Jean Ray. I have recently joined Agilex. My background is in bioanalytical as well as analytical for the last 40 years or so. I started out at Midwest Research Institute, now MRI Global. We were focused on supporting the National Cancer Institute for their anti-tumor and their antiviral programs. We established methods initially for how to validate and set the standards for that for the US government for all their programs. I then moved into bioanalytical, supporting all the toxicology trials. After getting my PhD in pharmaceutical chemistry, I then joined a CRO working for that company for about 30 years. My background was really in tissue analysis with focus on bioanalytical. I've also served on a IRB for the last 12 years. It's a phase 1, first in human trials, and I've been supporting that for the Western Copernicus group. Now that I'm with Agilex, I'm serving as the senior scientific advisor, providing technical support for Jason's group and their US operations.
Thanks, Jane. So our aim today is to give you an overview of sample collection and how some of that can impact your subsequent sample analysis. Now, you will hopefully gain insight into how these parameters could impact your bioanalysis drug program and help you hopefully ask more relevant questions of your clinical site and also your bioanalytical CRO. And we'll briefly touch on collection logistics, which can be especially relevant for patient trials. And then we'll give a brief overview of Agilex.
So let's start looking at sample integrity from the clinical perspective. The integrity of biological samples can often be compromised when the sample comes in contact with either a plastic or glass surfaces. The surface contact exposure is an important consideration throughout the sample collection procedures at the clinic, as well as the analysis procedures in the laboratory. The analytes in biological fluids that have low protein concentration, for example, urine, CSF, and bowel fluids, often bind to plastic sample containers, resulting in loss of analyte and quantitation errors. Charged analytes, typically the bases and the peptides, can similarly bind to glass. During method development, we find that the selection of the material for the designated sample container is often the most critical decision made in the development of the sample collection procedure. Examples of analytes with high nonspecific binding are listed here for both plastic and glass. Most notable is nitroglycerin, paclitaxel that bind both the plastic and glass, making it somewhat of a challenge for the laboratory to pick the appropriate container and container system for biological sample collection analysis. Of these, nitroglycerin is really a unique compound. Not only does it bind to plastic, but it dissolves into plastics itself. So in this particular case, nitroglycerin needs to be maintained in silanized glassware when collected and then analyzed wherever possible with silanized glassware in the laboratory. Agilex uses a Fivetube test to assess nonspecific binding of a given sample container. For example, when urine is spiked with a known concentration placed into a tube, it's then allowed to be vortexed and then incubated for a period of time to allow non-specific binding to occur if it is going to happen. At a given time, a portion of that sample is transferred to the second tube, again, vortex, allowed to sit for a period of time, and then a portion of that tube is then transferred to the subsequent tube all the way through tube 5. This is how we typically look at determination, the extent where possible of non-specific binding. Urine in particular is problematic in the fact that it does bind to sample containers because of the long exposure time during sample collection during a clinical trial. Of these additives that are fairly effective, Agilex finds that Triton X100 at 0.1% is the most effective, and it's sort of our go-to additive for minimizing non-specific binding. So here's an example where we looked at a small peptide, cyclic peptide, with two separate polypropylene tubes, A, standard polypropylene tube, and C, a low binding polypropylene tube that's commercially available from Eppendorf. And in each case, a parallel experiment was also done with the addition of Triton X, again at 0.1%, as noted in B and C. As you can see from the data, the addition of the Triton X was highly effective in minimizing the extent of non-specific binding for each of these particular tubes. Of the tube, the low bind tube was the most effective, but both met acceptance criteria for minimizing the degree of non-specific binding. The solubility of drugs in urine in particular can be exceeded when the samples are refrigerated during a collection interval, pretty common during a clinical trial, for example, a 0 to 4 hour collection. This results in the analyte falling out of solution, or we refer to as precipitation. To overcome this issue, organic solvents, for example, isopropanol, can be added to the collection container to then resolubilize the precipitate. The urine sample collection really only needs to be weighed, a specific volume of solvent added on a weight basis, and the container thoroughly mixed before aliquots are removed for subsequent analysis. This seems to work pretty well when this is an issue. For stabilization of samples at a clinic, whenever possible, we always try to use a specialty tube that's commercially available. Often, all we need is a buffer or some degree of acidification to stabilize a compound in the blood collection tube, such as the ACD tube or the acidic citrate tube that are commercially available. When this is not possible, the drug is highly unstable, a derivatizing reagent may need to be required to be added to the blood collection tube so it immediately comes in contact with the blood and stabilizes from that point forward. These are custom tubes, which I'll discuss later. Another tube, which is also commercially available and highly effective, is the sodium fluoride potassium oxalate tube. This includes an esterase inhibitor or so-called poison, which stabilizes esters from the degradation from the esterase enzyme. And lastly, there is a commercial tube, which is proprietary, called P800. It's often used for metabolic markers, but Agilex finds it's quite effective in stabilizing angiotensin in particular. At the clinic, though, occasionally you may need a more acidic addition, such as phosphoric, to stabilize compounds such as acylglucuronides further. This can be somewhat problematic because you need to add on a volume for volume basis. You also may need to add, in some cases, antioxidants, such as ascorbic acid, that are not available in a commercial tube situation. So these are things that we will request the laboratory to assist in collections. So let's look at an example of tubes that were evaluated in our laboratory for a notably unstable compound called an acylglucuronide. These are problematic for most bioanalytical labs in the fact that when they do hydrolyze, they result in an increase in the parent compound as the sugar moiety is released during the hydrolysis process, whether enzymatic or by hydrolytic reactions. In this particular data set, we looked at 4 separate tubes, EDTA, as well as the ACD tube and the sodium fluoride potassium oxalate tube that we discussed on the earlier slide. But also we included an additional tube called the homocysteine determination tube. Looking at the data, we found that the homocysteine tube, which is a fairly uncommon tube, was highly effective in stabilizing the acylglucuronide versus the other options we looked at. Again, with the homocysteine tube, the parent compound was stable with no elevation, which proved the tube worked well in stabilizing the metabolite. There are examples where thermally unstable compounds need to be evaluated for how to set up constraints during sample collections. That includes aldehyde, drug conjugates, esters, and lactones. Stabilization of plasma typically only involves the use of a chilled collection tube, refrigerated centrifuge, and immediately sample freezing during collection. However, for the really thermally unstable sensitive compounds, we might require to immediately freeze the whole blood sample immediately upon collection on a slant to keep brake tube from breaking. Most bioanalytical labs are designed with precautions in place to minimize the extent of compounds degrading due to UV light exposure. However, at a clinical site, this is not always the case. A lot of times we'll see that the exterior windows may not be properly covered, and samples can be exposed to light and then result in degradation. Here, the laboratory will try to work with the clinical sites to make sure that this is in place, so there's not an issue when transporting samples to the laboratory for the processing and subsequent placement into the freezer locations. Photodegradation can also be accelerated by free radical decay. The laboratory may suggest the addition of an antioxidant upon the collection of the samples, such as adding ascorbic acid that was mentioned before, or BHA, BHT to minimize analyte loss.
Now, sample analysis integrity. Now, to me, this means how do we ensure the concentration of drug as it comes out of the subject's arm, primarily in blood? How do we ensure that when we measure that weeks or months later in the bioanalytical lab, how do we ensure that that concentration in plasma is accurately reflective of that moment it came out of the subject's arm? And ultimately, this means that you can get quality data, and that then allows confident drug program decisions to be made. So to start that, plasma or whole blood. The decision of collecting plasma or whole blood for a study is determined knowing the extent of the drug partitioning into red blood cells or other cellular components like white blood cells and platelets. The recommended guide has been plasma for that ratio is sort of less than 1, and when whole blood when that ratio is greater than 1. And if plasma is used instead of whole blood in PK where the ratio exceeds 1, plasma clearance calculations can significantly overestimate drug clearance. Having said that, plasma is still generally accepted as the standard for PK measurements. Whole blood is generally only chosen when that ratio is significantly higher in blood, such as shown here for tacrolimus, where that ratio is 55:1, or methylazolamide, where that ratio can be 10:27, depending upon dose. Now, that partitioning can be affected by temperature, hematocrit, pH, or even the anticoagulant. And all of these are issues that can be controlled during that collection process. And furthermore, this is a reason to not necessarily default to storing your blood tubes, collected blood tubes on ice without knowing what that blood to plasma ratio might be and if that cooling temperature might impact that partitioning ratio. So to show an example of this, we have endoxifen here, which is one of the major active metabolites of tamoxifen, which is often used as a breast cancer treatment. And then endoxifen itself is being looked at now as a drug in its own right, as theoretically it may be more potent as it does not require that initial metabolism of tamoxifen for its activity. Now, endoxifen exists in both Z and E isomers, as depicted here, which is then rotation around that double bond. Now, this is one of our normal whole blood stability experiments, where as per our standard process, fresh whole blood is spiked with the analyte, endoxifen in this instance, incubated at 37 degrees for say 10 minutes, and then subaliquots are taken. And they're then exposed to the different stress conditions. So here, ice and room temperature for 30 or 60 minutes, and that's all referenced against the time zero. So at those different time points, The blood will then be processed as per what would be anticipated to be done in the clinic, and the plasma would be harvested and then subsequently analysed, in this case by supported liquid extraction. Now, as you can see here from the results, the ratio of Z to E in plasma is around 3:1, which is quite interesting. Further to note is there was no difference in this case between storing the blood on ice or at room temperature. Now, to further probe this and understand, is that difference in ratio a result of petitioning or potential instability of the E endoxifen, we then repeated the experiment in a slightly different manner, where the blood was prepared in the same way, subaliquoted as before, but then at those individual time points, instead of processing to plasma at, say, time 0 or after 1 hour, the blood was directly precipitated with solvent to effectively quench any reactions at that point, and then that supernatant was analysed. And this removes that harvesting of plasma process. So now you can see here in those numbers in red that the ratio between the Z and the E is now within, say, 15%, which is within analytical error. So this shows that it wasn't a stability issue in blood, whereby we would have lost E endoxifen, but an interesting case of differential petitioning of the E endoxifen into the cellular components in blood over the Z endoxifen. And this shows the importance of understanding what's happening during this process, otherwise erroneous or concentrations that you don't necessarily understand may be produced.
Now, there are examples where both plasma and whole blood need to be considered in the drug development and analysis procedures. Nucleoside drugs in particular are of interest, whether it's an antiviral or antineoplastic. Nucleosides can undergo rapid RBC uptake through the transporter uptake mechanism in cells. Once the nucleoside is incorporated within the cell, it can undergo phosphorylation, and the resulting phosphorylated adducts become trapped within the cell due to its high degree of ionization. Here, you'd be looking at both whole blood and plasma to have a true understanding of the degree of phosphorylation that might occur within the cells, as well as the degree of distribution within to the cells and elimination from the body. A really interesting area of drug research that started back in the 1980s is erythrocyte drug development, which also has a plasma and whole blood component for bioanalytical analysis for pharmacokinetic understanding. Here, packed red blood cells are placed in a hypotonic solution, which allows the cells to swell on the pores to allow easy partitioning of the drug within the cells as they're incubated, whether it's the drug itself or the drug in a formulation of nanoparticle. The matrix is then washed with an isotonic solution at a given time and results in a biocompatible drug formulation that can be reinfused into the subject with sustained exposure for the subject. As noted in the citations below, this is often used in oncology applications. Doxorubicin in particular is noted in the fact that these biocompatible drug formulations reduces the degree of cardiotoxicity of doxorubicin and minimizing doxorubicinol formation. So I think this is an area you'll see a lot more research, and one of our clients is particularly looking at this particular drug format.
The light sensitivity. Many drugs are highly sensitive to light, requiring samples to be processed under UV shielded laboratory light or in low light conditions. Now, the potential for photodegradation can cause oxidation, cis-transisomerism, and dimerization. Now, cis-transisomerism is like the example for endoxifen recently shown, where that E to Z ratio can be altered post-collection by white light exposure. So again, giving erroneous concentration data. Now, normally in the clinic, in whole blood, this isn't a problem due to the opacity of whole blood, which is good because controlling the clinic can be difficult, so they don't always have access to, say, yellow lights, for example. But amber plasma cryovials can be used if it is an issue. Now, many examples of drugs with this issue are known, such as fluphenazole, doxepin, and doxorubicin. where we have some data here to share, where doxorubicin, which is an anti-cancer drug, has been stressed here in plasma for 6 and 24 hours at normal light protected, and at 4 degrees in room temperature. And that's all relative to a baseline measurement where the sample is only stored at minus 70 degrees. So as can be seen here, the only real issue where instability of the drug is seen is 24 hours at room temperature and normal light. So what does that mean? That means then at the bioanalytical lab, all plasma sample handling is then performed under yellow light to control this light exposure and to not have any chance of instability seen. Now in the clinic, this didn't require any special conditions because the lab manual required from blood draw to plasma sample freezing was done within an hour. So as you can see here, that's well within the minus 6% loss of the drug. Now, controlling thermal degradation. An important step to maintaining sample integrity from thermal degradation is to always keep samples in a frozen condition over dry ice during both the sample inventory process, where the samples are received at Agilex Biolabs, and those samples are individually verified to ensure that we know the exact identity of each tube. And then subsequently, during that batch assembly process where the samples are arranged in a batch to then have the subject concentrations determined in an individual run, and during that verification process to ensure each tube is what we think it is. Now for labile compounds, samples should always be thawed in wet ice bath, and after pipetting, the residual samples may be immediately flash frozen using a dry ice acetone bath. Now temperature is one of the simplest control measures, which in turn makes it one of the best because it's safe, it's easy to use, and it's widely accessible. Now, thawed exposure at the bioanalytical site needs to be tightly controlled for traceability and proof of the validated test conditions. And that flash freezing, for example, can be really relevant for unstable compounds because a sample may be assayed 3 times for primary analysis, a repeat for some reason, and then if it's then chosen for incurred sample reanalysis. So that time can accumulate if it's not carefully controlled. Now, a related issue is the analysis of labile compounds in tissues, which has some extra challenges. Now, the simple process of homogenizing tissue prior to analysis can generate a significant amount of heat in that process, which then degrades the analyte of interest before you've even had a chance to measure it. Now, this can be overcome using chilled homogenization equipment, and pulsing samples can dramatically reduce analyte degradation. So the way we do that at Agilex is we commonly use it's called a bead beta homogenization. So that might have a 20 or 30 second pulse with the beads to homogenize the tissue, followed by a 2 to 3 minute cool down cycle where nothing happens to ensure that no heat builds up. And then if required, that process might be repeated 2 or 3 times. Alternatively, dry ice milling of tissues can be used. Now, enzymatic degradation. Biological samples can immediately undergo enzymatic degradation as they thaw and during that entire process. So since enzymes have optimum pH and temperature requirements, simple addition of an appropriate acid, base or buffer and maintaining the samples over wet ice may be sufficient for stabilisation. Now, a lot of this is why those commercial tubes exist, such as EDTA or the fluoride oxalate tubes to control some of these enzymatic processes. Now, another effective approach is to nature the enzymes by addition of an organic solvent to the sample. So this is protein precipitation. Now, this is not commonly used, but it can be really effective, especially in a non-clinical space, as there the processing staff often have a greater scientific background and more access to the appropriate equipment. It can be more difficult to process at a human clinical site due to the nature of the staff. Now, alternatively, an enzymatic inhibitor can be added to further stabilise the sample. And these can include aldehyde oxidase inhibitors, cytidine deaminase inhibitors, such as the well-known tetrahydrouridine, esterase inhibitors, such as sodium fluoride, which is in commercial tubes, dichlorobos, and then protease inhibitors. Now, this allows for a more targeted approach, but again, the selection needs to be careful to ensure it's not toxic to the collection staff. For example, dichlorobos can be quite hazardous. Now, in addition to the photothermal and enzymatic degradation we've discussed, integrity of the sample from chemical degradation is also essential to be controlled. Now, this can be through oxidation, hydrolysis, isomerization, racemization, and metabolite conversion to the parent drug. And that might be from acyl glucuronides, as Jean mentioned, or an N-oxide or other metabolites. And some of this can depend upon the drug structure itself and how it might be designed to work within the body. Now, an example of racemization that's probably well known is thalidomide, where the R enantiomer has sedative effects, whereas the S isomer is teratogenic. So under biological conditions, however, the isomers interconvert. So even if you dosed the R isomer only, it would interconvert into the S isomer in the body and thus cause issues. Now, oxidation. Several compounds are highly susceptible to oxidation, in particular aldehydes and thiols, and they can be controlled with the addition of antioxidants, such as ascorbic acid and BHT, sodium metabisulfite, which is actually quite well known for controlling the stability of catecholamines, such as epinephrine and dopamine. And then there's reducing agents like dithiothreatol, DTT. Now, hydrolysis. Acyl glucuronide metabolites are prone to hydrolysis even under frozen conditions at minus 70 degrees. Their hydrolysis cleaves the labile glucuronide moiety from the parent drug, which can impact ISR. So in the example on the right for ibuprofen acyl glucuronide, as Jean mentioned, that sugar moiety may be cleaved off, which then releases ibuprofen, which is now indistinguishable from free ibuprofen that was existing before that instability occurred. So this can have significant impacts. So, for example, when ISR testing fails with a notable increase in the reanalysis concentration of the primary analyte, this often suggests the presence of an acyl glucuronide in the sample. And this can be especially relevant in urine, where the acyl glucuronide may concentrate, and it might be 100 times the concentration of the free drug. So even a 10% degradation of that acyl glucuronide can have significant impacts on that free drug concentration. Generally, acid is used to control this, and a pH less than 5 is generally useful. Now, using that same acyl glucuronide as an example of isomerization, we can see from this picture, the top left is the normal beta-glucuronide that will be formed in a subject, which may then ring migrate to form the 2, 3, or 4 isomers, and then each of those isomers can then interconvert from the beta to the alpha isomer. So this is a stability issue and also an isomer issue in that, especially if we need to look at that acyl glucuronide drug and determine the concentration of it, we've now taken one moiety and potentially distributed to 6 different drug compounds, which may not be appropriately measured. And this may significantly impact the drug program if incorrect drug concentrations are given. Now, an example of this in our lab is shown here, where we're looking at acyl glucuronide metabolite in urine. So these are LCMS chromatograms. So on the left there, that blue triangular peak is that analyte at time 0 in pH 5.4 urine. Now we can stress that sample for 24 hours at that 5.4 pH, and we still see a single peak and no instability seen. However, in higher pH urine, shown on the right there at 7.9, which was unadjusted urine that was directly from a volunteer, after 24 hours, we can see multiple peaks have been formed. So that's a reflection of what was shown in the previous picture where isomerization has occurred. And so if this wasn't then controlled, we would clearly incorrectly give concentrations for that analyte. Now, also unexpectedly, this analyte suffered from non-specific binding, as did the parent, because normally you'd expect an acyl glucuronide to be much more soluble, and so thus not have problems with non-specific binding. So ultimately, the solution to this was we added trinate stabilizer, as Jenny previously mentioned, and also acetic acid to then control for the pH. So then both the pH and that non-specific binding was controlled in the urine samples to ensure accurate concentrations were produced. Now I might walk you through a case study example just to show a lot of these things in action. So this was the drug was a new chemical entity. The sponsor originally came to Agilex Biolabs to rescue their non-clinical method development program where the assay delays had stalled their tox study program. So they had animals, rat and beagle plasma, and it displayed instability. And we found in our labs, 1-to-1 stabilization of the plasma with ascorbic acid gave sufficient plasma stability. Now, as these samples are already 6 to 9 months old, when they came to Agilex, the plasma had already been collected, so we didn't have any opportunity to try and stabilize the blood. Now, the non-clinical samples were then successfully assayed at Agilex and concentrations reported to the sponsor. So that then causes the question, what about stability in the human matrix? So now this is a commercial blood tube evaluation. So this is where we looked at EDTA, lithium heparin, ACD, those homocysteine detection tubes previously used, which is an acidic citrate, and also rapid serum tube, just to try and assess the gross blood stability. And as you can see here by those ugly red numbers, we saw significant instability in whole blood, where after 30 minutes, at least 60% of the drug was gone. And interestingly, this stability was much worse than in the animal species. And we see slight differences between the two donors shown here, but in other data not shown, some donors almost had acceptable stability, which showed a significant difference between different donors. So we had to routinely test matrix for multiple donors to ensure we're looking at the potential worst case scenario to ensure we had control for that situation. And also the drug was much more stable at high concentrations where this instability was not really seen, such as say at 50 nanograms per mil. Now, to look at blood versus plasma, on the left table, we've spiked the analyte just into EDTA blood here and then stored on ice and stressed it for 30 and 60 minutes. Versus the right-hand table where that same analyte was spiked into EDTA plasma and stored on ice. So again, replicating the previous slide, you can see within 30 minutes, 70% of the drug is lost. However, in plasma, we certainly saw more stability where up to say 4 hours, we're starting to get acceptable stability. So this showed that we needed to focus on the blood to control the stability. And hopefully once we determine a solution for the blood, that would then help with the plasma. Now we looked at antioxidants to try and control this instability, but they didn't have sufficient impact. And also, we're also looking at a couple of metabolites of this analyte, and those antioxidants had differential impact on those analytes, so we couldn't use them as a single fix for all 3 analytes. Now, from enzyme activity work the sponsor had done, it appeared that their drug was a substrate of human aldehyde oxidase. Thus, we chose 3 AO inhibitors to test. Now here we show data from the hydrolysine, which was ultimately chosen because of its ease of use, certainly its effectiveness, and it was slightly less hazardous than some of the other options. Now, really pleasingly, we can see acceptable stability in whole blood, in an EDTA blood tube that then had hydrolysine at 0.75 milligrams per mil added. Now we can see this is both at 1 nanogram per mil and at 50, where 1 nanogram, we certainly were seeing gross instability up to minus 70, 80% within that 30-minute time frame. So the conclusion from this, a significant body of work was done to ensure analyte stability was controlled in both human blood and plasma. Custom commercial blood tubes are in preparation to support the human clinical trials and to allow consistent, simple, and well-controlled collection at that clinical site. And most pleasingly for the sponsor, their drug program can now progress to the human clinical study with confidence.
So this is a good point of looking at how important it is to establish a procedure which provides clinical trial integrity. It's important to have a strategy when determining the best solution to maintain sample integrity for these unstable drugs and biological fluids. The laboratory really needs to consider, most important, the constraints at a clinical site. A key consideration is to be able to maintain compliance with a formal collection procedure. We find that a two-stage approach is best. For example, with aspirin, a specialty tube, such as sodium chloride, potassium oxalate, would be specified by the laboratory for sample collections at the clinic to ensure stability for up to 10 days at minus 70. The laboratory, however, would need additional stabilization to extend that analysis window that would not be practical with the potentially hazardous reagent that might have to be employed at the clinic. So this two-stage approach really does work well when looking at the practicalities and also the compliance across the board for clinical trial management. In large clinical trials, in addition to ensure sample integrity, it can be logistically challenging given that there's a large number of sites and potential language barriers that are involved. A well-established procedure for establishing the collection procedure, along with steps that are used to mitigate the analyte degradation, are critical to sample trial success. When the addition of a stabilizer is necessary at a sample collection, the clinical team needs to also have calibrated pipettes to ensure that the proper volume of stabilizer is added to the samples. Whenever possible, we try to not require this particular step. Since there's so many sites involved, the degree of calibration necessary can be problematic. And so we try to keep it as simple as possible for compliance. Agilex has a kit production application as part of our agreements with our sponsors. We have a centralized kit production that's in conjunction with Markin. These kits that we provide are study-specific, site-specific, and visit-specific, and also contain pre-barcoded kits with draw tubes and aliquot tubes. The sample collection production is quite short, typically only about 6 weeks from the time of contracting, and these kits are automatically distributed and managed by Markin through its parent company, UPS for Agilex. We provide a sample kit laboratory manual. The sites are provided again with this comprehensive manual for the collection of samples. It outlines the sample collection procedures that we've gone through today, processing of samples, as well as packing and shipping requirements for the samples back to the laboratory. For replenishing supplies, our relationship with Markin only takes about 5 days to get those kits sent back to the site upon request. And in addition, we provide pre-printed air bills and the initial supplies that are sent to a site to ship these samples back to Agilex to make samples shipments easy for the clinic to send back to the laboratory.
And now I'll give a brief overview of Agilex Biolabs. Now, we've existed for about 20 years, so a strong history. We work with clients from around the world, primarily China, Korea, USA, Germany, and pharma companies within Australia. We've got more than 850 projects under our belt, and we really pride ourselves on our repeat customer rate of over 85%. We feel that once sponsors have worked with us and understand our strong customer focus, they're really comfortable working with us again. Now we have 3 broad groups: toxicology for in-life work, LCMS bioanalysis, where Jean and I are talking from today, and immunoassay and immunobiology for large molecules and biomarker work. And also Australia has a R&D tax incentive where potentially up to 43.5% of your spend for R&D conducted in Australia can be rebated. So that's for any bioanalytical work done in Australia. Labs, we're growing actively. We've got over 130 staff now, and many from around the world, which gives us a really broad knowledge base and a broad array of experiences. So we've seen a lot of different things and can approach things from a lot of different ways. Got 12 LCMS systems. So we use a SciEx platform. So we've got 3 of the really sensitive 6500 pluses, and then the more workhorse machines are the 4500 and the API 4000, all from SciEx. Now we've got over 300 validated biochemical assays, and most of these are primarily for new chemical entities and their metabolites, where all the stability discussion we've had today are directly relevant. So thank you for your interest in this today, and please feel free to contact Jean or myself. Now I'll pass back to Arsalan.
All right, thank you guys. Excellent presentation over there. Let's get to the audience questions now. Anyone in the audience, please just hit that Q&A button over there, and we will get that over to Jason and Jean. But I've got a couple questions for you, and let's get started. So first off, why is plasma and not serum used for PK sampling?
I'll take that, Arsalan. Most important, plasma is so much faster to process samples and get the resulting supernatant into the freezer. So that's kind of key. We always want to use plasma whenever possible. And again, ETA is sort of the go-to because heparin tends to get stringy over time. In addition, plasma typically generates more supernatant. So it also helps for pediatric trials. So that's also a consideration. And further, it can eliminate some degree of hemolysis for particular samples using plasma instead of a standard clot tube, red top tube.
Very good. Okay. Another question coming in here. So is it better to use commercial tubes than stabilizing the plasma?
Yes. For the simple fact it's simpler. It's simpler for the clinic to execute, and that means a higher rate of collection compliance, and that's what we're always interested in, and knowing that things are well controlled. Additionally, additives to blood can cause additional hemolysis, and if we don't need to complicate the process, we certainly don't want to and don't need to.
And so what is derivatization used for?
Yeah, Arsalan, derivatization is really important when you have a really unstable compound. The derivatizer reagent basically sets up a covalent bond with the adduct. So it's a stable bond and really locks the analyte in place for analysis. Sometimes the derivatization could also enhance detection. And so that could also be another reason why you use derivatization.
Okay. I just want to encourage the audience to keep coming your questions in over here for Jason and Jean. Let's continue with the questions over here. So why do you normally prefer Triton X100 to fix NSB?
I'll take that. Generally because it's effective. So we're always guided by the data. So if it's effective, it means it can be used. But also it can be cheap, especially compared to BSA, like bovine serum albumin, where potentially at the additive rate, it might cost $20,000 or $30,000 just for the BSA to control NSB in your urine. Also, it's easy to add. It's a liquid. It can be used at a low percent, like a 0.1% additive rate, as Jean mentioned. And it can be bought as a 10% solution. So it makes it really easy for the clinic to add it directly to urine rather than making solutions from powders, etc.
And for urine, do you need to add the stabilizer to the primary collection vessel?
Again, that's probably study specific. So ideally, if non-specific binding is not significant, we don't need to add it to that primary collection vessel, and it can be added to the pooling vessel for urine at the clinic to then be mixed thoroughly there, and then it helps control when that urine is then subaliquoted into the smaller vessels for the subaliquots, which then get sent to the lab. we have that stabilizer in there then, and that's normally in the smaller volumes where non-specific binding is more severe. However, if testing shows that we need to add it to the primary vessel, we will. But again, that may limit what we can use. And again, it needs to be simple for the clinic to be able to execute.
What are low bind tubes?
Low bind tubes are these commercially available tubes that Epanor produces. It's an ultra high purity form of polypropylene. And they also have some real constraints on the actual manufacturing of these tubes. It's just a modest increase, but in some cases it can be essential, as we noted here.
And for our final question here, what homogenizers do you recommend and why?
I'll take this one. This is a great one in the fact that tissues can be really difficult for a bioanalytical lab to work with. We find out for small sample sizes that the Precipitation works really well. This is a bead homogenizer. It gives us a lot of flexibility. You can use ceramic beads, you can use glass beads, you can use metal beads, and you can select the beads that work best, reduce the amount of non-specific binding. But more importantly, the Precipitation allows you to process samples in as little as 10 seconds. You can chill the unit the entire time, and that really can be a value. However, these do not work well for large sample sizes. So in that case, we recommend the Ultraturax or something along that line. Those are standard standalone homogenizers. They work really well, but they're also prone to contamination from one sample to the next. So it takes a lot of extra effort and validation in knowing how to best clean the probe from one sample to another. So that's a consideration in looking at which direction you go.
Well, very good. That's all the time that we have for today, Gene. Gene Ray and Jason Goy, thank you very much. And thank you to everyone in the audience for tuning in. And we appreciate Agilex for sponsoring the discussion at Endpoints webinars. If you'd like to rewatch today's session or to share it with your colleagues, a link for on-demand viewing will be sent tomorrow. I'm Arslan Arif for Endpoints News. Thank you for joining us and hope to see you at a future webinar event.